Now, there's over 400,000 different species of flowering plants, so we know they're a very, very successful type of organism. Now, we need to know some reasons why they're so successful. Well, one of the first reasons is that if we've got deciduous plants, that means that these plants shed their leaves once a year, and the next year they kind of develop new leaves. Now, when the leaves get shed and released from the plant, we know they fall to the ground, and we know they get decomposed. So we've got decomposer organisms, so decomposition, and that helps to release some useful molecules or ions into the soil, things like nitrate ions or phosphate ions. Now the roots of these plants can then reabsorb these inorganic ions like nitrates and phosphates, and they can use them and assimilate them back into the cells. So it's like a recycling of very useful material by the plant. Now we also know that these flowering plants have a very rapid life cycle. So once the flowers emerge, we know very, very soon after that point we get successful pollination of the flower. So the pollen grain gets transferred into the flower, and we know we get successful fertilization and seed formation. So the point from which the flowers open to the point whereby we get seed formation is very rapid. Now we also know once those seeds have been formed, inside the seed not only is there the embryo that will develop into the new seedling later on after germination, there's also food reserves to allow that embryo to develop so rapidly. Now if it's a monocotyledon plant, the food reserves will be in the form of endosperm, and if it's a dicotyledon plant, the food reserves will be in the seed leaves, or the embryo leaves if you like, called the cotyledons. So once that seed starts to germinate, there's a ready-made food reserve inside the seed that provides the glucose that's needed to be absorbed by all the cells of the embryo to allow it to respire and therefore develop very rapidly. Now the other point is that the seed, once it's released from the flower, so before it germinates, we know it can withstand very adverse conditions because we know it's got a very hard outer coat called the tester. Now the tester withstands things like UV radiation. So if the seeds are wind dispersed, they might go quite high up into the atmosphere and they might be exposed to high levels of UV radiation. That would cause damage to the DNA inside the cells inside the seed, but this tester helps to withstand the UV radiation. We also know the tester protects the seed from environmental conditions like frost, for example. So we could say protection from frost. Now we also know that dispersal of these seeds, whether it's by an animal, so if the seeds contained within a fruit, quite often these are eaten by animals, but the animal can't digest the seed because of the tester, so they get dispersed quite far away from the original parent plant, or the seeds might be dispersed on the wind, so wind dispersed seeds. In either case, these seeds are distributed far away, so we could say far away from the parent plant. Now that's going to allow the seed, once it germinates, to develop into a seedling. The seedling will develop roots and it will absorb mineral ions from the soil like nitrates and phosphates, and there's a lack of competition from the parent plant because the seedling develops so far away from it. Now we also know that these flowering plants have evolved a very close relationship with pollinating insects such as bees or wasps, for example, or other pollinating animals like birds. So they will aid in seed dispersal if it is a flowering plant that has brightly coloured petals, for example. So we also need to know a little bit about the structure of a flower. Now this flower we know is going to be animal pollinated. So the animal could either be an insect like a bee or a wasp, or it could also be a bird, or it could be another animal whereby the pollen inside the anther is transferred onto the fur of the animal as it brushes past the flower. Normally a certain type of flower, a certain species of flowering plant will have a certain species of pollinator. So there's quite a close relationship between the species of flower and the actual species of the pollinator. Now we'll start with the female part of the flower. So the female reproductive organ is called the carpel. And this elongated part of the carpel is called the style. And on top of the style, we get a structure called the stigma. Now the stigma is quite sticky and it will bind pollen grains if we get successful pollination. Now quite often if the pollen grain lands on a compatible stigma, the stigma secretes sucrose solutions which can start to stimulate germination of the pollen grain. Now the rounded structure at the bottom of the carpel is called the ovary, and within the ovary we can either have, depending on the species, a single ovule that contains the female egg cell, i.e. the female gamete, or we can have multiple ovules inside the ovary. Now each ovule, if there's multiple ovules, will contain a single egg cell. So in this particular scenario here, we've got 6 ovules inside the ovary, and each ovule will have an egg cell that will then get fertilized if we get successful pollination. Now the receptacle is this green structure at the bottom of the flower, and it's basically the upper part of a stem that helps to keep the flower in the correct orientation to allow successful pollinators to enter the flower to get to the nectary. So the receptacle is a green colour and it's the top part of the stem. Now the sepal is the green. They look like leaves and they do photosynthesize to a certain extent on the outside of the flower. Now these help to protect the flower when it's inside the bud, and then the flower emerges and the petals start to emerge, and the sepal will be seen at the bottom part of the flower. So they can actually photosynthesize because they do have the chlorophyll pigments in the thylakoid membranes of the chloroplasts inside the cells of this particular tissue. Now we know the petals are very brightly coloured. Normally they have a certain orientation to allow, if it's insect pollinated, the insect to land on the petal. We know at the bottom of the petals are nectaries, so we can put nectary, and we know the nectary contains nectar, and that basically is a sugar solution that if it's an insect pollinator they can feed off, so the bee will drink the nectar and take it back to the beehive. Now the male part of the flower is the anther. Now the anther contains the pollen grains. So we're going to see later pollen grains are made from what we call micro-spore mother cells. So pollen grains are actually a type of spore, and inside those we're going to have the male gamete nuclei form. So inside the anthers we get lots and lots of pollen grains that contain the male gamete. Now this elongated green structure is called the filament, and that basically allows, it contains vascular tissue like xylem and phloem tissue that supplies the anther with things like sucrose and amino acids from the sieve tubes and water and mineral ions from the xylem vessels. So the previous slide we did show an animal pollinated flower. Now we also get wind pollinated flowers. So these will not have brightly coloured big petals with the nectary and nectar. Instead what we do see is the carpel, the female reproductive organ. We know it has an ovary, which is this rounded structure here, and inside it is normally a single ovule. So inside this we will have the ovule that contains the egg cell. Now we can see the style here is this elongated green structure, and there's actually 3 of them in this case. And projecting out from the style, we don't have a single stigma in this case, we actually have multiple stigmas. And they look like a feathery structure to increase the surface area. So we've got multiple stigmas, so we can actually see them on this photo on the right hand side. And they have a very large surface area because of the feathery like appearance. And the point of this is that the stigmas attach pollen grains. Now if the pollen grains are distributed on the wind, we know there's less chance that the pollen grain gets transmitted from a different flower onto this wind-pollinated flower, because it's quite random. So we have to have a very large surface area to catch the pollen grains being distributed in the wind. So for example, if we had some pollen grains up here in the wind, they would have to randomly attach to these sticky stigmas with a very large surface area, very feathery structure. Now we can also see the stamen, which is the male reproductive organ, the stamen, made of the anther and the filaments. Now in this case, the anther is not inside the flower itself, but it hangs outside the main part of the flower. So for example, here in the photograph, the anthers would hang low outside the main part of the flower. And we know the filament contains xylem vessels and also phloem tissue, so things like sieve tubes and companion cells, and they supply the anther with water and mineral ions and also sucrose and amino acids through the phloem. So we can have some quick recap questions then. Well, where does meiosis occur in a flower? Well, there's two areas. One is the anther, and that produces pollen grains. So we know micro-spore mother cells undergo meiosis, and that's going to form immature pollen grains. We know in the ovary we have one or more ovules. Now we know inside the ovule we will have meiosis, and that will form an embryo sac, and inside the embryo sac we will have the egg cell, so the female gamete egg cell. So we've got an anther, is the male part of the flower. We've got an ovary, which is the female part of the flower. Now, some species of flowering plants, the flowers can be either male, whereby you just find the anther inside them, or they can be female, where you get the carpel with the ovary with the ovules inside, or in some cases, the anthers and the ovaries, or the carpel we should say, is part of the same flower. Now, what's pollination then? Well, it's the successful transfer of pollen grains from the male anther of one flower to the female stigma of the carpel of a different flower of the same species. So successful transfer of pollen grains from the male anther of one flower to the female sticky stigma of the carpel of another flower of the same species. Now we know pollen grains, when they get released, either attached to an insect or on the wind, they have to be protected, and on the outside of a pollen grain is the structure called the exine, and that prevents drying out of the pollen grains, and it also prevents damage from UV radiation, for example, because pollen grains don't necessarily, if it's a wind pollinated flower, get transferred straight from the male anther to the female stigma. They could blow in the wind, they could land on different structures, and it's quite random if they end up actually successfully pollinating a different flower. Now, what's double fertilization in flowering plants? Well, we're going to see later, when the pollen grain lands on the stigma, the pollen tube emerges and grows down the style of the carpel. Now, there's a nucleus inside the pollen tube, and that nucleus undergoes mitosis to form two male gametes. And when these male gametes reach the embryo sac that we'll do shortly, we actually get a double fertilization event. So one of the male gametes will fertilize what's called the central cell, and there's two haploid nuclei inside this. So we get a haploid male gamete nucleus, and that fuses with two polar nuclei, and that will make a triploid nucleus called the endosperm, or the endosperm cell, that divides to form endosperm tissue. Now, the other male gamete nucleus fertilizes the egg cell, and that produces a diploid zygote. So a diploid zygote, and it's this diploid zygote that divides by mitosis to form the embryo that's contained within the seed. So what happens to the fertilized ovule? Well, it actually becomes the seed. So the outside of the ovule has layers of tissue called integuments, and the integuments on the outside of the ovule form the tester of the seed. The fertilized egg cell inside the ovule becomes the diploid zygote, which becomes the embryo inside the seed. And if it's a monocot plant, the central cell with the triploid nucleus becomes the endosperm. So if we look at the cross-section of an anther in a bit more detail, what we'll find is 4 structures called pollen sacs. So these structures in brown here are called the pollen sacs. Now inside these pollen sacs we're going to get formation of pollen grains, inside which we will have the male gamete. Now there's a layer of tissue shown in green here called the tapetum. Now the tapetum provides nutrients, which might be glucose, amino acids, nitrates, phosphates, and other chemicals and hormones, which stimulates and nourishes the developing pollen grains. So the layer in red is a layer of cells called micro-spore mother cells, and these are diploid starting cells. Now the micro-spore mother cells undergo what we call meiosis. So we're going to start with a diploid, so 2N micro-spore mother cell. That undergoes meiosis 1, so meiosis 1, and that forms 2 haploid cells. Both of the haploid cells then undergo meiosis 2, and we end up with 4 haploid cells, and that's called a tetrad. So the starting cell is a micro-spore mother cell, it's diploid. It undergoes meiosis 1 to form 2 cells that are haploid. They both undergo meiosis 2, and we end up with 4 haploid cells. Now in this picture you can only see 3 cells, but they are all attached to each other in a sphere. So there's actually 4 cells in this structure at the bottom. Now each of these 4 cells, called microspores, they then develop into immature pollen grains. So immature pollen grains. So meiosis occurs from the microspore mother cells. We end up with a tetrad of pollen, and they each individually develop into immature pollen grains. Now this is the immature pollen grain on the left hand side. So it's an immature pollen grain. Now we know it has a haploid nucleus. That means it's got one set of chromosomes inside this nucleus structure inside the immature pollen grain. Now this nucleus undergoes mitosis. So the DNA inside the nucleus will replicate, and then we get nuclear division. So we end up with 2 nuclei inside the immature pollen grain. Now the outside of this pollen grain then starts to develop into what we call the exine. So one nucleus inside the immature pollen grain divides by mitosis after DNA replication. We get 2 nuclei inside the pollen grain, both haploid. We get an outer exine layer, which is very, very tough, and it's got spikes on the outside. And inside the exine, we get another thin layer called the intine. Now, it's important just to notice that these 2 haploid nuclei, they've got certain names. One of them is called the generative nucleus, and one of them is called the pollen tube nucleus. Now, what we're going to see later is it's the generative nucleus, so this one here on the left-hand side in blue. When we get successful pollination, the generative nucleus, when it enters the pollen tube after the pollen grain germinates, it undergoes mitosis again. So it's the generative nucleus. Inside that we have DNA replication to double the DNA content. The nucleus then divides by mitosis. So we end up with 2 haploid nuclei in blue at the bottom, and these are both male gamete nuclei. So we've got 2 male gamete nuclei, and we also obviously have the pollen tube nucleus, which is also haploid. Now this microscope image shows a cross-section of an anther, and you can actually see the pollen sacs here that would have contained or do contain the pollen grains. Now, along the length of an anther from top to bottom are lines of weakness, and at certain times of the year these lines of weakness will rupture, and this is called dehiscence. So dehiscence, and we call these the lateral grooves. So the lateral grooves are a line of weakness along the length of the anther from top to bottom. This is a cross section that rupture, and that releases the pollen grains from the pollen sacs to the outside, whether that be attachment to an insect if it's an insect pollinated flower, or released on the wind if it's a wind pollinated flower. Now, these pollen grains that you can see here in the pollen sacs that get released inside, so if we draw one up top left, if that was inside the pollen grain, we know there would actually be two nuclei inside this. So the purple one might be the pollen tube nucleus, and we know that's haploid, and that's going to control the growth of the pollen tube when we get successful pollination. And the other nucleus is the generative nucleus, so that's also haploid. Now, the generative nucleus is going to divide by mitosis to produce two male gamete nuclei. That doesn't really occur until the pollen grain has been released and it attaches to the stigma of a flower on a different plant of the same species, and that pollen grain starts to germinate and the pollen tube emerges. So when the generative nucleus goes down into the pollen tube after successful pollination and germination of the pollen grain, then the generative nucleus divides by mitosis to form the two male gamete nuclei. Now, we also need to talk about formation of the egg cell inside the ovule. So inside the ovule, we're going to get the egg cell, and that is the female, so the female gamete. And we know that's going to be haploid, so a single set of chromosomes. Now, this structure up top left here is called the carpel, and that's the female reproductive organ inside a flower. So we know the top is the stigma. We know the elongated structure through which the pollen tube will move is the style. Now, the ovary is this kind of protective rounded structure at the bottom of the carpel, and inside the ovary we find an ovule. Now, if it's a monocot, so monocot or monocotyledon plant, like the grasses, they're very simple plants, one ovary will contain one ovule. Now, in the dicotyledon, so a dicot plant, dicotyledon, there can be multiple ovules within a single ovary. So this ovary here in the picture top left has two ovules inside it, so we know it's a dicot plant or dicotyledon plant. Now, inside an ovule then, we have a structure called the embryo sac. So this round structure is called the embryo sac. Now, the layers of tissue on the outside of the ovule are called integuments, and they will actually form the outside tester of the seed if we get successful fertilization. Now, the cell inside this ovule is called the mega-spore mother cell, and that's a diploid cell, so two copies of each chromosome, diploid. And the mega-spore mother cell undergoes meiosis, so it's going to form four haploid, genetically unique megaspores. So the mega-spore mother cell, which is diploid, is the starting cell to form the embryo sac. That is diploid. It undergoes meiosis 1 and then meiosis 2, so we can put that here. So meiosis 1 and 2. We know at the end of meiosis we get four genetically unique haploid cells, so they're genetically unique, and they're called megaspores. Now, of these four cells, only one of them will develop. So the four haploid megaspores, one of them will continue its development, so it gets bigger and it makes more organelles, and it's going to have quite a high metabolic rate, and the other three cells die off. So we call this the mega-spore. There you go. Now that haploid nucleus inside this mega-spore cell, that's going to undergo 3 rounds of mitosis. So the DNA inside that nucleus, there's one set of chromosomes, the DNA replicates, and then the nucleus undergoes mitosis. So we'll end up with one mega-spore with 2 haploid nuclei inside because there's no cytokinesis. Those 2 nuclei will each undergo mitosis. So we've gone from one nucleus to 2 nuclei. Each of these nuclei is then divided by mitosis to form 4 nuclei, and we get another mitotic division, and we get 8 haploid nuclei inside this mega-spore cell. So 3 rounds of mitosis. So if we start to look inside the ovule, so this is the embryo sac here at the bottom. We've got a single cell, and we've got 1, 2, 3, 4, 5, 6, 7, 8 haploid nuclei. So they're all haploid nuclei. Now this embryo sac here at the bottom, it's now going to undergo cytoplasmic division or cytokinesis. Now there's not 8 cytokinesis events, there's actually 7. So what we end up with inside the ovule is 3 haploid cells at the top called antiploidal cells. So they're called antiploidal cells, and they're all haploid. We get 3 haploid cells at the bottom. So these in green are called synergids. So synergids at the bottom of the embryo sac, synergids, and they're also haploid. Now the cell right in the middle, the haploid cell, is called the egg cell, this one here at the bottom. So that is a haploid egg cell, and that is the female gamete. Now the central cell, so this cell right in the middle here, it's called the central cell. We can see that has 2 haploid nuclei, and they're called polar nuclei. So polar nuclei, and there's 2 of them inside that central cell. So we've got 7 cells, they're all haploid apart from the central cell, which has got 2 haploid nuclei, so it's actually diploid. Now the antiploidal cells, the 3 of them at the top, they die off and disintegrate, so they don't do anything. The 2 synergids either side of the egg cell also die off, and that leaves inside the mature embryo sac a single central cell with 2 haploid nuclei and an egg cell with a single haploid nucleus, and the egg cell is obviously the female gamete. Now when the pollen tube develops down the style tissue, the pollen tube nucleus disintegrates as the pollen tube grows up into the ovule. So the pollen tube will grow up through the micropyle this way into the ovule. Now one of the male gamete nuclei will fertilize the egg cell, and that forms a diploid zygote, and the second male gamete nucleus, the haploid one as well, will fertilize the central cell, and that will produce a triploid endosperm cell. And these endosperm cells will increase in number through mitosis, and that forms the tissue that ends up being the food reserve inside the seed. So we know the integuments on the outside of the ovule develop into the tester of the seed. We know the egg cell, it's haploid. Once it gets fertilized by a male gamete nucleus, it forms a diploid zygote. The diploid zygote divides by mitosis to form the actual embryo that will form the new plant. And the central cell with its 2 haploid nuclei gets fertilized by a second male gamete nucleus, and that forms a triploid cell, which divides by mitosis to form the endosperm tissue, which is the storage of starch inside the seed. So we can look at a slightly different diagram here. It's the same idea. So nucellus is a layer of tissue on the outside of what will become the embryo sac. So actually, if we do that in blue, you can see here, this is the nucellus in the original yellow layer. Now that basically makes and secretes chemicals that help nurture the embryo sac and help its development. Now the integuments on the outside, these layers of tissue on the outside of the ovule, we know if we get successful fertilization or double fertilization, they will form the tester of the seed coats, so the tester of the seed coat. We know at the bottom of the ovule is a gap called the micropyle through which the pollen tube inserts, and that allows the two male gametes to get inside the ovule during fertilization. We've got one single cell at this point called the mega-spore mother cell, and that's diploid. And it's this cell that's going to undergo meiosis. And when it does so, we know the diploid mega-spore mother cell forms four haploid megaspores. So we can call these megaspores. Now, 3 of them die off. So one of these cells, maybe this one at the top, the haploid cell, will continue to get bigger. And it's still haploid, remember. And it will undergo 3 rounds of mitotic nuclear division. So we go from 1 haploid nucleus, in this case to 2. Each of them undergoes another mitosis, so we end up with 4 nuclei. And then if each of these undergoes another mitosis, we're going to end up with, if we just draw them all in, let's go back there, 8 nuclei. So 1, 2, 3, 4, 5, 6, 7, 8 nuclei inside this cell. Now we then get cytokinesis, and that gives us 7 cells. The 3 cells at the top, the antiploidals, die off. So we can see here with these crosses, they die off. The 2 synergids die off. That leaves the central cell in the middle, this one with 2 polar nuclei, and the egg cell, the haploid egg cell at the bottom. So this is the actual embryo sac that would be surrounded by the integuments on the outside of the ovule. So we can look at this sequence of events with some possibly slightly clearer diagrams. So let's have a look at what the notes are going to be. So diploid mega-spore mother cell undergoes meiosis 1 and 2. We get 4 haploid cells. Of the 4 haploid cells, one of them gets larger. That's the mega-spore cell that's going to continue to develop. The other 3 degenerate. The mega-spore cell that gets bigger, the single haploid nucleus undergoes 3 rounds of mitotic division, so we end up with a single big megaspore cell with 8 nuclei inside it. Cytokinesis then occurs, we get cytoplasm division, we get 1 central cell and 1 egg cell. All the other haploid cells die off. So the megaspore mother cell is diploid, that's the starting cell that will end up forming the embryo sac. We get 4 haploid cells, one of them becomes the megaspore cell, which is also haploid. We get enlargement of the megaspore cell. We get 3 rounds of mitotic division. We get 8 haploid nuclei, and then we end up with 3 antiploidal cells that die off and degenerate. We end up with 2 polar nuclei inside a central cell. We end up with 1 egg cell, which is the female gamete, and it's haploid, and the 2 synergids, they also disintegrate. Now, we also need to know a bit about pollen grain formation. So we know pollen grain formation occurs inside the pollen sacs of the anther. Now, we know there's a layer of tissue on the inside of the pollen sac, and that will give rise to what we call micro-spore mother cells. So we said earlier that the cell that forms the egg cell inside the ovule is the mega-spore mother cell. Well, this is the micro-spore mother cell, so the micro-spore mother cell in this case. Now we know this is going to undergo meiosis, so this here must be meiosis 1, and we get 2 haploid cells being formed. Now each haploid cell undergoes meiosis 2, so we're going to have 4 cells being produced, all genetically unique and all haploid. So let's put in some of the answers then. So meiosis 1 at the top, meiosis 2 underneath that, and we're going to get a tetrad of microspores. So we call these micro-spores. So the original starting cell was the microspore mother cell. And here we've now got 4 haploid microspores. So if we looked at the chromosome number, well, we know, imagine this was 40 at the top, so it's diploid. The chromosome number after meiosis 1 would be halved. So we go from diploid, 2n, to haploid. So it's a halving of the chromosome number from 40 to 20. Now we know meiosis 2 is very similar to mitosis. So we know all 4 cells are haploid and there's 20 chromosomes this time as well, but the chromosomes inside the nucleus at the end of meiosis 1 will still be replicated chromosomes made of a pair of sister chromatids. The chromosomes inside the nuclei at the end of meiosis 2 are single chromatids, which are now called sister chromosomes. Now, these microspore cells separate, and each microspore cell becomes an immature pollen grain. Now, the haploid nucleus inside each immature pollen grain undergoes mitosis. So we have one nucleus, one haploid nucleus inside an immature pollen grain called a microspore. This undergoes mitosis to make 2 haploid nuclei inside this microspore. We then get cytokinesis, so we end up with 2 cells really inside this microspore or immature pollen grain. One is called the generative nucleus, which is haploid here, and the other one is called the pollen tube nucleus, both haploid. So the pollen tube nucleus is haploid, the generative nucleus is haploid. This generative nucleus, once we get successful pollination and the pollen tube forms, will actually divide by mitosis again to form the 2 male gamete nuclei, which we know are both haploid. Now, the outside of the immature pollen grain then, well, it forms 2 layers. The inside layer is called the intine, this kind of lighter sort of orangey colour, and the outside layer is called the exine. Now, this allows protection of the genetic material inside the cell or inside the pollen grain. It also quite often has spikes and hooks of a characteristic pattern on the outside, and that allows attachment to insects for pollination if it's an insect pollinated species of flower. It also prevents drying out, so the pollen grain does not dry out. It protects against UV damage, protects against conditions like frost as well. Now, self-pollination then. Well, this is really where a pollen grain is produced from an anther and it successfully lands on the sticky stigma and then germinates on the female reproductive organ, the carpel, inside the same flower. So pollen is transferred, as we just saw, from the anther onto the sticky stigma of the carpel of the same flower. Now, one type of flowering plants that we get self-pollination in, or we can see self-pollination, are the orchids. So we can see here, these are the anthers, they make the pollen, they release pollen grains, and they land on the sticky stigma, and they can germinate, and they will then allow several pollen tubes to grow down the style, and each pollen tube will deliver 2 male gamete nuclei inside the embryo sac of the ovule, and we will get double fertilization. So each ovule inside the ovary will form a seed. So if we had successful pollination of 1, 2, 3, 4, 5, 6, so we could put here 1, 2, 3, 4, 5, and 6 ovules, so we'd have a minimum of 6 pollen tubes going down the style, we'd end up with 6 seeds inside this ovary. So we know this must be a dicotyledon plant because the ovary has 6 ovules, which all can be fertilized by double fertilization. Right, cross-pollination then. Well, that is the transfer of a pollen grain from the anther of a flower on one plant to the sticky stigma of a carpel inside the flower on a different plant but of the same species. So we saw here the pollen grains are produced by the anther. They go and they get transferred by whatever means onto the sticky stigma of the carpel within a different flower of a different plant but of the same, so of the same species. So there you go. Now, this is quite highly beneficial because cross-pollination does ensure the mixing of genotypes and it increases genetic variation within offspring. Because the gametes, i.e. maybe the pollen grains produced from this individual, so the pollen grains are all unique, produced by this individual plant, they will be very different from all the egg cells, the genetic material inside the egg cells, produced by the other. So the egg cells of the other individual. So the egg cells we know are within the ovule, within the ovary, or ovules we could say. So how do flowering plants then prevent self-pollination? Well, it depends when either the anthers mature compared to when the stigma of the carpel matures. So in this case, on the left-hand side here, we can actually see the anthers have matured and dehiscence has taken place. So the pollen grains have been released into the atmosphere or onto the back of an insect or an animal. But we don't get self-fertilization because this stigma here, as part of the carpel, this whole reproductive organ of the female, the stigma is not fully matured. So these pollen grains that have just been released can't land on and germinate on this particular stigma. Now, afterwards, once the anthers have kind of withered away and died, then the stigma matures. So that promotes pollen grains from different flowers on different plants of the same species being released and binding to the sticky stigma. And it kind of limits any pollen grains from these anthers within the same flower self-pollinating. Now, so we call that Protandry, and that's where the male anthers mature before the female stigma. The other mechanism is called Protogyny here on the right. So if you think of gynecological as female possibly, so Protogyny, that's where the stigma, so this structure here in blue, that's the sticky stigma, that matures first within this flower before the anthers have matured. So the pollen grains are still maturing inside these anthers, not quite there yet. So pollen grains from flowers on a different plant of the same species might be released, they might come in, bind to the sticky stigma, we might get successful pollen grain germination. Only after this time do the anthers of this particular flower then mature and they release their pollen grains into the atmosphere like so. But by this time the sticky stigma of the carpel in this particular flower has degraded somewhat, so these released pollen grains are not going to land and germinate on the stigma within the same flower. That's called Protogyny. So in this slide we're going to talk about the germination of the pollen grain. So once a compatible pollen grain lands on the sticky stigma of the carpel inside the flower of or on a different plant, it will germinate. Now it can absorb water from the stigma, and the stigma also releases certain sugars like sucrose, which gets absorbed by the pollen grain, and that germinates to start to make a pollen tube, and that pollen tube grows down the style tissue and through the wall of the ovary and eventually up through the micropyle. Now the pollen tube nucleus is always at the very tip of the pollen tube. Now that nucleus has genes inside the nucleus that are switched on, and these genes will make or code for proteins that are hydrolytic, so we can put hydrolytic enzymes. Now these hydrolytic enzymes are secreted from the tip of the pollen tube, and they digest the style tissue and the ovary wall tissue as the pollen tube moves down. Now the breakdown products of this digestion are absorbed by the pollen tube, and they allow it to continue its development and growth towards the ovule. Now this is called chemotrophism, because the embryo sac inside the ovule, so if I just highlight the ovule as this bit here, we can see the integuments are on the outside that will form the tester. The embryo sac is inside the ovule, and that releases chemicals to attract the pollen tube towards it. So this big elongated tube down the style tissue, down the wall of the ovary, is attracted to the ovule through chemicals released by the embryo sac called chemotrophism. Now the generative nucleus, as it passes down the pollen tube, that will divide by mitosis, and that will form two male gamete nuclei that are both haploid. So once the pollen tube grows through the micropyle into the embryo sac, the pollen tube nucleus disintegrates, the tip of the pollen tube bursts, the two male gamete nuclei, so I'll highlight those in orange down here, so that's one here and that's the second one. So the two male gamete nuclei enter the embryo sac, and because these three cells have disintegrated at the top and the two synergids have disintegrated, we've got a single, do that in blue, central cell with the two polar nuclei, and we have a single egg cell with a haploid nucleus. Now we get two fertilization events. One of the male gamete nuclei will fuse with the central cell, and the other male gamete nucleus will fuse with the egg cell. So we end up with a triploid. So I can put this here, triploid endosperm cell, which divides by mitosis to form endosperm tissue. And we end up with a diploid, so a diploid 2N zygote. And that again will divide by mitosis to form the embryo plant inside the seed. So the ovule becomes the seed, the integuments on the outside of the ovule become the tester, and the embryo sac with the central cell and the egg cell, that will become the endosperm, which is triploid, and the embryo of the seed. So this is a summary of the process that takes place in double fertilization. So you can just read through this. The pollen grain lands on the stigma. It can absorb some water, and it can absorb certain types of sugars that allow it to germinate. The pollen tube grows down the style tissue. We know the pollen tube is chemotrophic. That means it grows towards the embryo sac within the ovule, because the embryo sac releases chemicals that attract the pollen tube towards it. We know the pollen tube nucleus contains genes that are switched on that will code for hydrolytic enzymes that get secreted from the tip of the pollen tube to help digest the style tissue. Products from that breakdown are absorbed by the pollen tube to allow it to elongate. Now, the two male gamete nuclei are formed from mitosis of the generative nucleus. The pollen tube then enters through the micropyle of the ovule, and it passes into the embryo sac. The pollen tube nucleus then disintegrates, and the tip of the pollen tube then bursts. That releases the two male gametes, or the two male gamete nuclei, into the embryo sac, and then we get double fertilization. One of the male gamete nuclei, which is haploid, will fuse with and fertilize the central cell that contains the two polar nuclei, and that will form a triploid cell that divides by mitosis to form the endosperm. The second male gamete nucleus will enter and fuse with the embryo cell nucleus to form a diploid zygote. That diploid zygote will then divide by mitosis to form the actual embryo plant inside the seed. So once we get successful double fertilization inside each ovule, what happens to the ovary then? Well, the ovary itself actually starts to develop into a fruit. So this structure here on the right hand side, this is the fruit wall. So the fruit develops from the ovary. Now each ovule on the left hand side. So we can see we've got 3, 4, 5 ovules here. If they are all successfully doubly fertilized, we'll end up with 1, 2, 3, 4, 5 seeds on this side. Now we can see we've also got 2, 3, 5 ovules on the left hand side here. So again, each ovule will develop into a seed. So we'll have 5 seeds this side. So this fruit, for example, might have 5 seeds inside it. So the ovary originally would have had 5 ovules inside it. Now, apple trees, for example, the flowers of apple trees have a carpel, and inside the ovary of the carpel, there's normally 10 ovules. Now, if each ovule becomes fertilized, or the embryo sac inside the egg cell becomes fertilized, each flower will develop into an apple, so that's the fruit. Inside the apple will be 10 seeds. Now, sometimes when we get things like nuts, it's actually the a modified fruit wall forms the outside of the nut, and the seeds can be found inside the nut structure. Other times, the fruit is very succulent, like citrus fruits, apples, etc., and that promotes being eaten by certain animals. Now, we know the tester of the seeds are quite resistant to digestion, so they don't get digested and broken down inside the stomach and the ileum of the animal, and they get egested out further away from the parent plant. The further away the seeds are from the parent plant, when they germinate into a seedling, there's less competition for resources in the soil, things like nitrates, phosphates, water, etc., sunlight as well. So it's going to cause less intra-specific competition. So you can see the ovules, once they're fertilized, they become the seeds. We can see the ovary. After we get successful fertilization, that becomes the fruit. Now, 2 types of flowering plants. One are the Monocotyledons, and these are quite straightforward, simple grasses. Things like maize and wheat are also Monocotyledons. So inside the seed, what we'll see is this big structure here is the triploid endosperm tissue. So triploid endosperm tissue. Now, the embryo is this structure on the right hand side. So this is what the diploid zygote develops into, this kind of embryo structure. Now, part of the embryo, called the plumule, that becomes the shoot of the seedling once the seedling emerges and starts to develop from the germinated seed. Now, the radicle is actually the root system of the embryo. Well, what will form the root system of the seedling once the embryo starts to develop even further, once the seed germinates. Now, this structure within the embryo is called the cotyledon. So a monocot, the word mono, the prefix mono suggests one, and cotyledon means embryo seed leaf. So there's a single seed leaf as part of the embryo here. Now, in a monocot, the endosperm contains the starch stores that will be needed for release of glucose or alpha glucose from maltose, which is the disaccharide that gets cleaved off the amylose and amylopectin of starch. Those sugars are needed in respiration inside the cells of the embryo to release the energy in the form of ATP to allow cell division. Now, dicots are a little bit more complicated. Things like the broad bean here. We can see the ovary contains 3 ovules. If we get successful double fertilization inside each ovule of the embryo sac, we will actually have 3 seeds within the fruit structure. So here we've got 3 seeds. This structure on the outside would have been the ovary, and now it's developed into the fruit. So broad beans you can think of as modified fruits, if you like. So this is the what's left over from the ovary after it's kind of developed. Inside you can actually see the 3 seeds. So each of these would have been a single ovule. So it would have been a single ovule with a single embryo sac inside. But inside each of these seeds here, so we've got 3 seeds. it's slightly different to the monocots. So again, we've got the embryo inside the seed that will form the seedling after seed germination. We've got a radicle and a plumule like we had before, but di means two, and we know cotyledon means seed leaf. So there are two seed leaves here, and the endosperm, so the big kind of starch stores, they get absorbed into the two cotyledons. Now, the whole job of these cotyledons is that when the seedling starts to emerge from the seed, once it germinates, and the shoot breaks through the soil, so the plumule breaks through the top of the soil, these cotyledons, the starch stores within them get used up initially to allow further development of the seedling until the first true leaves are formed, which can then photosynthesize and make glucose for further development, because we know glucose is used in aerobic respiration to make ATP, which is needed for cell division. Now, in some species of flowering plants or dicots, the two cotyledons or seed leaves that emerge first, they are actually a green color and they can photosynthesize, but in others they're a kind of browner color, and the starch stores just get used up and they then disappear. So a dicotyledon, such as a broad bean, the seeds, the embryo inside the seed will have two seed leaves called cotyledons. The food store in the endosperm gets absorbed into the cotyledons. So the embryo will have two seed leaves, and they contain lots and lots of starch stores as well. We know the fruit is formed from the fertilized ovary. We know the embryo plant inside the seed has developed by mitosis from the diploid zygote. We know the tester, the outside of each seed, which is very resistant to things like UV irradiation, frost, and drying out of the seed. That forms from the integuments of the ovule, and the seed itself, the whole complete structure, formed from the ovule. So if we talk a little bit about seed germination then, well, we know the seeds are located in the soil, normally maybe a few inches below the surface of the soil. Now, there are several things needed for the seed to germinate. One is the correct oxygen levels, because we know oxygen is needed for aerobic respiration in the cells of the embryo inside the seed. We also need the correct temperature, because the enzymes inside the embryo cells have to work optimally at that temperature, and also water availability or moisture of the soil is very important, because we know water enters the seed through the micropyle, and there are imbibed, so it's called imbibing or imbibed, where the water enters the micropyle into the seed. Now, that water is going to stimulate the cells of the embryo to secrete a hormone called Gibberellin, and Gibberellin stimulates the cells of the endosperm, or the cells of the embryo itself, to make amylase, which digest starch to maltose. So the seed, once it takes in water, the outside tester is going to kind of get a little bit thinner, and it's going to kind of be shed as the seed starts to emerge as a seedling. So we know the radicle of the embryo, that's going to develop into the root system of the seedling, and we know that grows down towards gravity. We know the root hairs of the root system absorb water, and we know that's needed for the seedling to continue to develop. Now, the part of the embryo that develops into the shoot of the seedling is called the plumule. So that's the part of the embryo inside the seed that will develop and grow outwards and upwards against gravity to form the shoot. So we can see here, this bit at the top, that would have been the plumule of the embryo, so that's the shoot system. And we know plants grow upwards vertically. Now, the two seed leaves are the cotyledons, and there's two of them. So we can see them here. They're the brown looking leaves. Now, these contain high levels of starch. So this would have been the endosperm tissue that was absorbed by the embryo, the two embryo leaves called the cotyledons. Now here inside these leaves, these cotyledon leaves, the starch stores are digested by the production of amylase. We know amylase breaks down amylose and amylopectin. So amylopectin, and that forms maltose. So the maltose gets translocated throughout the seedling, going up and down. So going up and down the sieve tubes as part of the phloem tissue. And we know maltose will actually again be hydrolyzed to alpha glucose, and that's what's used inside the cells for aerobic respiration to make the ATP. Now the cotyledons supply the maltose and therefore the glucose for aerobic respiration, but when these stores are used up, quite often the cotyledons will just kind of wither and die off, and the first true leaves start to emerge here in green. And because they are green, we know they have chlorophyll A, which is one of the main photosynthetic pigments that reflects green light. So we know these can photosynthesize and make glucose, and that is therefore obviously going to be translocated up and down the seedling to the shoot tip and the root tip as sucrose quite often. And the glucose will be used in aerobic respiration in predominantly in the cells of the plant that need the most ATP, i.e. those that are undergoing high levels of cell division. Now, we said earlier there are three conditions needed for germination of seeds: correct oxygen levels, the correct temperature so the enzymes work optimally, and the correct water moisture. Now here we've got several different tubes that have been set up. Inside each tube is a culture media or liquid media with a certain level of moisture within it, so a certain volume of water would have been added. So this one here and the two on the right hand side. The second tube here we can see has not had any water added to it, so this would be without water. So we can see the effect on germination, and we can see the seeds have not germinated here. In tube number 1, where the temperature is suboptimal, so it's only 4 degrees C as opposed to say maybe 25 degrees C, the seeds have not germinated because that's not the optimum temperature. 4 degrees C, it's too cold. And the presence of oxygen, when there's no oxygen in this tube in the middle, you can see here no oxygen, the seeds do not germinate. Now you can add certain chemicals that absorb oxygen from the air within the tube, and if that oxygen is absorbed, then there's no oxygen to be able to be taken in by the cells, the embryo cells inside the seeds, so obviously they can't respire aerobically. So a lack of oxygen prevents aerobic respiration in the cells of the embryo, and they can't make sufficient ATP. Now, if we look at the two tubes whereby we have germination of the seeds, well, we can see oxygen is present in this tube here. We can see the temperature is the correct temperature, so maybe 25 degrees C in this case, and we can see there's sufficient moisture or water content in the culture media or the soil, depending on what you want to use, to allow the seeds to germinate. So inside these seeds, if these were dicotyledon plants, we know the embryo cells would start to secrete gibberellin. We would get amylase being produced eventually, and any starch stores within the cotyledons will be broken down to maltose, and maltose can be broken down further to glucose, and that's used in, as we said, aerobic respiration. Now, anytime you do this type of experiments, there were certain control variables that you'd have to keep consistent. So if we're going to look at the seeds themselves, so you can see the seeds down here on the left, you've actually got 4 seeds here. Now you'd have to use 4 seeds in the second tube, and the seeds would be of the same species, so the same species of seed, and also the same size of seed. Now, this seed is going to represent what we call the monocot. So monocotyledon seed. So we know here the endosperm tissue still exists. So all this tissue here is called the endosperm tissue, and we know endosperm is triploid cells that make up the tissue. Now in green, inside the seed, you can see the embryo itself. Now part of this will be the plumule, which will develop later on into the shoot of the seedling. Part of this embryo will be the radicle that will develop into the root system of the emerging seedling. Now we know when water enters the seed through the micropyle, so there is actually a little hole in the tester. So a little hole in the tester, and we know water can enter that. It gets imbibed into the seed. That stimulates the cells of the embryo to secrete gibberellin, which is a hormone called gibberellic acid. So the entry of water activates the release of gibberellin from the embryo cells. That diffuses to the outside layer of the tissue inside the seed, and that's called the alerone layer on the outside. So this is within the seed, but the outside layer is called the alerone layer, and that stimulates the cells of the alerone layer to make proteases, to upregulate how many proteases are present, and that breaks down protein inside some of these cells to release lots of amino acids. And these amino acids are used in protein synthesis in the cells of the alerone layer to make more and more amylase, and that's a vital enzyme in this breakdown of the starch stores. So we know amylase that's secreted from the alerone layer cells breaks down the amylose and the amylopectin of starch inside the endosperm cells to release maltose. So gibberellic acid or gibberellin is a hormone that stimulates the alerone cells to make proteases. These hydrolyze any proteins inside these cells to release amino acids. This allows more amylase enzymes to be produced because we know amino acids are the subunits of enzymes. The amylase then breaks down the amylose and amylopectin stores inside the endosperm cells, and that's going to release maltose. Now the maltose is broken down further into alpha glucose because we know maltose is a disaccharide, and we know it's made from two alpha glucose joined together via an alpha 1 to 4 glycosidic bond. We know maltose and alpha glucose are absorbed, number 5 here, by the cells of the embryo. We know glucose is needed by every cell of the embryo to undergo aerobic respiration. We know the end product of that from the mitochondria inside these plant cells is ATP. We know ATP is needed for cell division, and that's needed to allow the embryo to divide by mitosis and get bigger and emerge from the seed as a seedling. The plumule grows upwards into the shoot system of the seedling, and the radicle grows downwards under gravity into the soil to form the root system of the seedling to help absorb nitrates, phosphates, mineral ions, and obviously water.